The groundnut, generally known as peanut, and the goober is taxonomically classified as a legume crop grown mainly for its edible seeds. It is widely grown in the tropics and subtropics, being important to both small and large commercial producers. It is classified as both a grain legume and, because of its high oil content, an oil crop. Atypically among croplands, peanut pods develop underground rather than above ground. It is this characteristic that used to assign the scientific name hypogea, which means under the earth. Vernacular names in South India. Peanuts are known by many other local names such as earth nuts, ground nuts, goober peas, monkey nuts, pygmy nuts, and pig nuts. Despite its name and appearance, the peanut is not a nut, but rather a legume. In Hindi and in South Indian languages, peanut is called moongpali in Hindi, kadalekai or nelakadale in Kannada, nilakadalai or verkadalai in Malayalam and Tamil, and verusanaga in Telugu. Origin. Cultivated peanut, Arachis hypogea, evolved from a hybrid between two wild species of peanut thought to be Arachis duranensis and Arachis ipensis. Genetic analysis suggests that hybridization event probably occurred to give rise to Arachis monticola, a wild form of peanut that occurred in a few restricted locations in northwestern Argentina, and Arachis hypogea was developed by artificial selection from this. From this primary center of origin, cultivation spread to Peru, Ecuador, Brazil, Paraguay, and Uruguay. Over time, thousands of peanut land races evolved. These are classified into 6 botanical varieties and 2 subspecies. Subspecies Arachis hypogea fastigiata types are more upright in their growth habit and have shorter crop cycle. Subspecies Arachis hypogea types spread more on the ground and have longer crop cycles. The oldest known archaeological remains of pods have been dated at about 7,600 years old. They were found in Peru, where dry climatic conditions are favorable to the preservation of organic material. Geographic distribution. World's annual production of shelled peanuts was about 42 million metric tons in 2014-15. The top 10 countries in production were China with 133.37 lakh metric tons, India 71.56 lakh metric tons, Nigeria 27.56 lakh metric tons, United States of America 18.37 lakh metric tons, Sudan 13.99, Indonesia 12.74, Myanmar 8.41, Senegal 6.94, Argentina 4.63, Vietnam with 4.15 lakh metric tons. In India, the Kharif groundnut crop of 2016-17 is estimated at 54.80 lakh metric ton against 32.30 lakh metric ton in previous year, increased by 70%. The top 10 states of peanut production are 1. Gujarat with 4,930,000 metric tons with the share of 50.77%. 2. Andhra Pradesh 1,230, 12.69%. 3. Tamil Nadu 960, 9.91%. 4. Rajasthan 910, 9.39%. 5. Karnataka 660, 6.81%. 6. Maharashtra 330, 3.41%. 7. Madhya Pradesh 200, 2.06%. 8. Uttar Pradesh 90.93%, 9 Odisha 0.83%, and 10 other states 310,000 metric tons and has share of 2.06%. Major peanut markets in India are at Rajkot, Ahmedabad, Mumbai and Delhi. Economic importance. Groundnut and its products have wide variety of uses as roasted nuts and proteinaceous products. Groundnut oil and butter are good cooking material. Byproducts like cakes and meals or animal feed components contains 40 to 50% protein, 6 to 12% fat and traces of vitamins. Groundnut milk acts as supplementary source to cattle milk. Variety of biscuits and cookies are also prepared from groundnut. Soil and climatic requirement. Soil. The best type of soil for groundnut cultivation is the one which is well-drained, light-colored, loose, friable, sandy loam, well supplied with calcium and moderate amount of organic matter. Soils with poor drainage, high acidity or alkalinity must be avoided for groundnut cultivation. A soil pH of over 5 or below 8.5 is supposed to be an ideal for groundnut production. The optimum temperature for growth of groundnut is between 27 degrees Celsius and 30 degrees Celsius. Climate. Groundnut is predominantly a crop of the tropics, but it is now grown throughout the tropical and subtropical countries and continental parts, that is warm parts, of the world. The approximate limits of present commercial production are between latitude 40 degrees north and 40 degrees south. It is grown up to an altitude of 1050 meters. Groundnut has been found to grow and yield well in areas receiving 40 to 125 centimeters of well-distributed rainfall. Varieties. Groundnut growing zones of India are, owing to the vast agroecological situation in which Kharif groundnut is grown, the entire area has been divided into 6 zones, of which Zone 5 and 6 are South Indian zones. Zone 5 is Peninsular Zone, which include Karnataka, Andhra Pradesh and South Maharashtra. The varieties recommended for Andhra Pradesh are, the number of varieties released are 4. Name of the varieties are ICGS 11, Kadiri 2, Kadiri 71-1, Kadiri 3. Varieties recommended for Karnataka. The number of varieties released are 6. Name of the varieties: Improved S206, DH330, KRG1, DH8, S230. Zone 6, that is Southern Zone. The varieties recommended for Tamil Nadu and Kerala. Number of varieties released are 16. Names of the varieties are MV1, TMV2, TMV3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Then POL1, 2, CO1, CO2. Classification of groundnut varieties. The cultivated groundnut, Arachis hypogea Linn, has 3 distinct botanical groups, namely Pish, subspecies Fastigiata, variety Vulgaris, Valencia, subspecies Fastigiata, variety Fastigiata, and Virginia, subspecies Hypogea hypogea. The Pish and Valencia types, popularly called as bunch, grow erect, possesses light green foliage and produce pods in clusters at the base of the plant. The seeds are non-dormant and roundish with light rose testa, deep rose or purple testa in Valencia. The Virginia group, which includes both Virginia bunch, that's semi-spreading, and Virginia runner, spreading type, on the other hand, possesses dark green foliage and the branches trail on the soil surface either partially or completely. The main stem is devoid of fruit, and pods are scattered all along the branches. The seeds are dormant, oblong in shape, with brownish testa. In general, the spreading and semi-spreading varieties mature late as compared with the bunch varieties. Plant description. Peanut is an annual herbaceous plant growing 30 to 50 cm tall. Like most other legumes, peanuts harbor symbiotic nitrogen-fixing bacteria in their root nodules. The leaves are opposite and pinnate with 4 leaflets, 2 opposite pairs, no terminal leaflet. The leaves are nystagnatic, that is, they have sleep movement, closing at night. Peanut pods develop underground an unusual feature known as geocarpy. The flowers are 1 to 1.5 cm across and yellowish orange with reddish veining. They have borne asexually clusters on the stems above ground and last for just 1 day. The ovary is located at the base of what appears to be the flower stem but is actually a highly elongated floral cup. After fertilization, a starch stalk at the base of the ovary, termed as pedicel, elongates to form a thread-like structure known as a peg. This peg grows down into the soil and the tip, which contains the ovary, develops into a mature peanut pod. Pods are 3 to 7 cm long, normally containing 1 to 4 seeds. Cultural practices. Field preparation. Plow the land till all the clods are broken and a fine tilth is obtained. To tide over the surface crusting, apply lime at 2 metric ton per hectare along with FIM or composted coir pith at 12.5 metric ton per hectare. Application of fertilizers. Apply NPK fertilizers as per soil test recommendation. If soil test is not done, follow the blanket recommendation. N25, P50, K75 kg per hectare and sulfur sludge 60 kg per hectare. N and K in 3 splits, viz. 50% N and K as basal plus 25% N and K at 20 days after sowing plus 25% N and K at 45 DAS is recommended. Forming of beds. Form beds of 10 square meter to 20 square meter depending upon the availability of water, slope of the land and type of soil. Ridges and furrows may be laid at 60 cm spacing. Sowing is done on both sides of the ridge alternately. Raised bed with width of 60 cm and a furrow of 15 cm on either side may be formed and sowing taken on the raised bed. Application of micronutrients. Apply TNAU micronutrient mixture at 12.5 kg per hectare as enriched FIM. Prepare enriched FIM at 1:10 ratio of MN mixture and FIM. Mix at friable moisture and incubate for 1 month in shade. Broadcast evenly on the soil surface immediately after sowing. Do not incorporate the micronutrient mixture to the soil. Seed rate. Use 12.5 kg per hectare of kernels. Increase the seed rate by 15% in the case of bold seeded varieties. Spacing. Adopt a spacing of 30 cm between rows and 10 cm between plants. Wherever groundnut ring mosaic bud necrosis is prevalent, adopt a spacing of 15 into 15 cm. Seed treatment. Treat the seeds with Trichoderma viride at 4 gram per kg of seed or Pseudomonas fluorescens at 10 gram per kg of seed. Biocontrol agents are compatible with biofertilizers. First, treat the seeds with biocontrol agents and then with rhizobium. Fungicides and biocontrol agents are incompatible. Sowing. Dibble the seeds at 4 cm depth along with fertilizers. Weed management. Pre-sowing. Flucloraline at 2 liter per hectare soil applied and incorporated following by light irrigation. Pre-emergence. Flucloraline 2 liter per hectare or pendimethalin at 3.3 liters per hectare applied on third day after sowing through flat fan nozzle with 500 liters of water per hectare followed by irrigation. After 35 to 40 days, one hand weeding may be given. Earthing up. Accomplish earthing up during second hand weeding. It is an important operation in groundnut. Earthing up is to be done within 40 to 45 days after sowing as it helps for the preparation of pegs in the soil and also facilitates for increased pod development. Do not disturb the soil after 45th day of sowing as it will affect pod formation adversely. Nutrient spray. Pod filling is a major problem, especially in the bold seed varieties. To improve pod filling, spraying of nutrient solution is to be given. This can be prepared by soaking DAP 2.5 kg, ammonium sulfate 1 kg and borax 0.5 kg in 37 liters of water overnight. The next day morning, it can be filtered and about 32 liters of mixture can be obtained and it may be diluted with 468 liters of water and made up to 500 liters of water and can be sprayed. Water management. Schedule the irrigation at 0.40 and 0.60 IW per CPE ratio during vegetative and reproductive phases respectively. Regulate irrigation according to the following growth phase of the crop. Pre-flowering phase 1 to 25 days, flowering phase 26 to 60 days, maturity phase 61 to 105 days. Crop protection. Pests. Many pests affect groundnut crops such as red hairy caterpillars, Amsecta mureri, Jacids, Empoesca kerri, groundnut leaf miner, Aproerema modicella, thrips, Certothrips dorsalis, Bihar hairy caterpillar, Pylosoma obliqua, jewel bit, Spinoptera indica, gram, pod borer, Helicoverpa armigera, termites, Odontotermes species, tobacco caterpillar, Spodoptera litura, white grubs, Holotrichia serrata, groundnut bud borer, Anarcia ephipias, pod borer, Anisolabes stalli, aphids, Aphis crassivora, podbug Aphaenus sordidus. These pests can be controlled by 1. Mechanical control methods. Collect and destroy egg masses and early instar larvae. Install pheromone trap at 5 per hectare of mass trapping. Spray neem-based formulation at 5%. The biological control methods are release Trichogramma chilonis or suitable agents 50,000 per hectare twice 7 to 10 days interval. Conserve the natural biocontrol population of spiders, long-horned grasshoppers, praying mantis, robber fly, ants, green lacewing, damselflies, dragonflies, flower bugs, shield bugs, ladybird beetles, groundnut beetles, predatory cricket, earwigs, NPV, green muscular fungus. Mulching with rice straw causes reduction in leaf miner incidence and increase in percentage of parasitism. Intercropping groundnut with pearl millet enhances the parasitide development. Chemical control methods. Apply safe chemical insecticides at recommended dose only if the insect population crosses the economic threshold limit. Effective control could be achieved if insecticide is applied at 45 and 70 days after planting. Carbaryl 50 WP 0.2% spray was found to be most economical for controlling most of the insect pests, or spray phenolphos 25 EC, 2 ml or methyl dimeton 25 EC, 1.6 ml or dimethoate 30 EC, 2 ml per liter of water. Diseases. Early leaf spot, Cercospora arachidicola. Symptoms. Small chlorotic spots appear on the leaflets with time enlarge and turn brown to black and assume subcircular shape on upper leaf surface. Management. Spray any one of the following: carbendazim 500 gram per hectare or mancozeb 1000 gram per hectare. If necessary, give the second round 15 days later. Late leaf spot, Fusariopsis personatum. Symptoms are black and nearly circular spots appear on the lower surface of the leaflets. Lesions are rough in appearance. In extreme cases, many lesions coalesce, resulting in premature senescence and shedding of the leaflets. Management. Intercropping pearl millet or sorghum with groundnut at 1:3 ratio is useful in reducing the intensity of late leaf spot. Deep burying the crop residues in the soil, removal of volunteer groundnut plants, and important measures in reducing the primary source of infection. Spray carbendazim 0.1% or mancozeb 0.2%. Rust, Puccinia arachidis. Pustules appear first on the lower surface, may be surrounded by colonies of secondary pustules. Later, pustules may also appear on the upper surface of leaflet. Spray mancozeb 1000 gram per hectare if necessary. Repeat the spray 15 days later. Stem rot, Sclerotium rothschild. Symptoms: pustules appear first on the lower surface, and in highly susceptible cultivars, the original pustules may be surrounded by colonies of secondary pustules. Pustules may also appear on the upper surface of the leaflet. Management. Seed treatment with Trichoderma viride at 4 gram per kg seed and soil application of Trichoderma viride at 2.5 kg per hectare mixed with 50 kg of FIM is recommended. Cropping systems. Row intercropping of peanut plus sorghum or ragi or red gram or chili or cotton or sunflower or castor, etc. in different proportion can be grown. Mostly mixed crop with pulses such as groundnut wheat, rice groundnut, rice groundnut. Economic uses. Following are some of the important uses of groundnut. Edible oil, 50% oil can be obtained from seeds. Haulms are palatable fodder to cattle, used as hay or silage. Groundnut kernels have high protein, 1.3% times higher than meat, 2.5 times higher than egg, 8 times than fruits. Oil has numerous industrial uses such as preparation of vegetable oil, vanaspati, low quality oils for soap, for beauty aids, shaving cream, cold cream, for medical aids, plasters, ointments, cake powder for laundry starch and manufacture of paper, gummy tapes and plastics, activated carbon, oil cake, good organic manure and cattle feed. Cake flour is easily blends with wheat and others to bakers, confectioners, candy makers and ice cream manufacturers. Harvesting and yield. Drying and falling of older leaves and yellowing of the top leaves indicate maturity. Pull out a few plants at random and shell out the pods. If the inner side of the shell is brownish black and not white, then the crop has matured. Irrigate prior to harvest if the soil is dry, as this will facilitate easy harvesting. Strip off the pods from the plant. Groundnut stripper developed by TNAU can be used. Dry the pods in sun for about 4 to 5 days. Repeat drying for 2 or 3 more days after interval of 2 or 3 days to ensure complete drying. When temperature is very high, avoid direct sun drying. Collect the pods in gunny bags and store on the ground over a layer of sand to avoid any moisture coming in contact with dry pods. Groundnut yields about 2000 kg per hectare bunch type and 3000 kg per hectare in spreading type varieties. Summary. The groundnut, generally known as peanut, and the goober is taxonomically classified as a legume crop grown mainly for its edible seeds. It is classified as both a grain legume and because of its high oil content and oil crop. Atypically among croplands, peanut pods develop underground rather than above ground. It is this characteristic that used to assign the specific name hypogea, which means under the earth. Despite its name and appearance, the peanut is not a nut, but rather a legume. In Hindi and in South Indian languages, peanut is called moongpali in Hindi, kadalekai or nelakadale in Kannada, nilakadalai or verkadalai in Malayalam and Tamil, and verusanaga in Telugu. Cultivated peanut, Arachis hypogea, evolved from a hybrid variety, Arachis monticola, a wild form of peanut that occurs in northwestern Argentina by means of artificial selection. The oldest known archaeological remains of pods have been dated at about 7,600 years old. They were found in Peru. World's annual production of shelled peanut was about 42 million metric tons, and the top countries are China, India, Nigeria, and United States. In India, the top producers of groundnut are Gujarat, Andhra Pradesh, Tamil Nadu, and Rajasthan. Sandy loam soil with pH of over 5 or below 8.5 and optimum temperature of 27 to 30 degrees Celsius is optimum for growth of groundnut. It yields well in areas receiving 40 to 125 cm of well-distributed rainfall. The cultivated groundnut Arachis hypogea has 3 distinct botanical groups, namely Pish, Valencia, and Virginia. Groundnut yields about 2000 kg per hectare in bunch type and 3000 kg per hectare in spreading type varieties. Thank you.